In this video, we will discuss about how the liver formation occurs in the prenatal stage within the woman from the, you can say, monocytes. So it begins. This is the embryonic vasculture. So at E7 stage, there is a formation of yolk cell or erythromyeloid progenitor cells, and it then differentiate into the pre-macrophages, and this happens at E7 stage, and after it forms into the pre-macrophages. It then generate at E8.5. These macrophages can migrate into the fetal tissue, where they can differentiate into the tissue resident macrophages. And these first tissue resident macrophages that they differentiate into is in the form of this liver, and that is shown here, that how it is formed. So a portion of these cells are still the yolk cell macrophages, and some of them are fetal liver, that is derived from homeopathic stem cells macrophages. And this thing occurs at E8.5. After that, at the stage of E9.5, what happens is this homogenic endothelia interacts with this EMP, and it will then directs this premature macrophages and monocytes into the blood, and it can then go to the other part of the body except brain, and that is shown here. And after that, so this thing happens at E9.5. After that, what happens, this homeopathic stem cells will also migrate into the spleen and bone marrow, where they are maintained as a progenitor cells for adult monocyte or further monocyte or macrophages development. And at this stage, the fetal liver is been developed. So additional things that we need to know, that how these progenitor cells differentiate, or know that they need to differentiate into the liver or other parts of the body, and these things can be done through the tissue specific transcription regulation. And some of the regulations might lead to, or differentiation or regulations might lead to this bone marrow formation, or the liver formation, or this choroid plexus, kidney cells, kidney tissue, skin tissue, or intestine. And these progenitor cells are usually has these self-renewal properties. And this at E10.5, what happens on that E10.5? What happens is that the HSC cells, this is homeopathic stem cells. This whatever the fetal liver that is formed so far is then seed into the homeopathic stem cells, and then it will then go into the resident macrophages. So that thing that I have forgot to inform you before, that at E10.5, whatever the fetal liver that is formed is then seed into the homeopathic stem cells, and after that these P macrophages and monocytes can move into the blood, and that can travel across all the tissues except brain. And these homeopathic stem cells then after this postnatal stages migrate to the spleen and bone marrow, where they are maintained as progenitor cells, that I have already said. So this part is usually occurs after the postnatal stage. And what happens after the birth is that these HSC derived bone marrow monocytes and early probe cells contribute to the tissue resident macrophages pools in some organs. So this tissue resident macrophages, the function of this tissue resident macrophages is that it will act as a part of the immune system, where if there is any harmful foreign objects or harmful things observed by the immune system of the body, then macrophages can be recruited at that site, and it can engulf those foreign or harmful proteins or harmful virus, and thereby prevent the cells from the prevent the cells from prevents the host body from being get damaged. So this is how this fetal liver formation begins or occurs within the at the prenatal stage within the woman. So I hope you got a basic idea in brief how this fetal liver formation occurs. If you need to understand in detail, about these different waves, that is yolk cell EMP derived, and this fetal liver EMP derived, and then third one is HSC derived. You can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.